Okay, so before we launch into talking about the FC area of the antibody in detail, I kind of wanted to remind you where that is. So the FC is the constant area of the antibody. So it is the long portion of the Y, okay, as opposed to the two arms of the Y, which are the variable regions. So the heavy or the constant region, or the FC region, is the part that binds to the macrophages and other cells. So if you look at those little green circles at the bottom of the cartoon, that is where the macrophage would bind to the antibody. Okay. So that FC end that we just were talking about can bind to receptors on the membranes of certain cells. So, such as macrophages, neutrophils, eosinophils, mast cells, basophils, and lymphocytes. So all different sorts of white blood cells. Now, the effect of the FC binding on the cell membrane depends on the cell role. So if the cell is a phagocyte, like a macrophage, then the antibody is related to something called opsonization, which is the attachment of the antibody to that foreign cell, and that is followed by the binding of the FC end to a phagocyte, which then engulfs the entire thing and kills whatever it is that the antibody is attached to. However, if it's an IgE that is attached to that particular toxin or pollen or whatever it is, it binds to basophils and mast cells, and those are both involved in allergy mediation. And so what that does is it causes the release of allergic mediators like histamine. And so that one class of antibodies, which we're going to talk about in just a couple of minutes, is different than the other four classes of antibodies. Okay, so there are 5 different classes of immunoglobulins, okay? And there are, one of those is referred to as IgA, okay? That is one of the isotypes. So it is, the IgA actually has 2 different forms that it can take. It can either be an individual antibody, which circulates in very small amounts in the bloodstream, or it can be a dimer, which means 2 of them attached together. And the FC ends are attached, and then the 2 Y ends are facing outward. And in a couple of slides, we're going to have a chart, and you'll see what that looks like. So that dimer is the one that lives in the mucous membranes and also in various different body fluids like saliva, and in the intestinal tract, in the mucus, in the nasal membranes, in breast, lung, and the genitourinary tract. Okay, so that dimer, as I mentioned, is 2 monomers that are held together by what's called a J chain. And there's a piece that gets added to that facilitates the transport of that IgA across membranes, because remember, IgA is a normal component of a lot of mucous membranes, and particularly ones that have a large moisture component, like for example in the mouth you have saliva, like on the eye you have tears that kind of coat the eye as well. Okay, so IgA coats the surface of mucous membranes because those, of course, are exposed mostly to outside air. Those IgA dimers are suspended in saliva, tears, colostrum, mucus, and it provides immunity against enteric, respiratory, and genitourinary pathogens. And just I wanted to point out the colostrum is the earliest secretion of breast milk, and it is very, very high in IgA, which in turn coats the gastrointestinal tract of a nursing infant. So remember that infants are born with virtually no immune system. And so in the mother's milk, there are these IgA antibodies which provide temporary immunity based on the things that the mother is immune to. So those IgA antibodies provide the baby's very first immunity until it can build up its own immune system. Okay, so as I mentioned, there are 5 different classes of immunoglobulins, and we're going to talk about them basically in order from left to right of the most common to the least common in the blood. Okay, so the most common is IgG. Okay, it is a monomer, which means it goes around by itself, sort of floating around in that little Y shape. Okay, it is about 80% of the antibodies floating around in the bloodstream. So this is the major antibody that is produced. It's also the most long-lived. So its average half-life in days is about 23 days, which means on average, it's going to live about 46 days. So the half-life means half of them have died by 23 days. It will cross the placenta. In fact, this is where we get into the problem with Rh negative mothers carrying an Rh positive fetus, because that is something that the mother's immune system will react to. And very often when there's a baby in utero, there is some passage of blood between the mother and the baby through the placenta. And that is why the Rh negative mom having an Rh positive baby is so much of a problem, because the mom's antibodies will in fact cross the placenta and attack the baby's bloodstream because it recognizes that as foreign. The FC, that bottom of the Y, attaches to phagocytes. So again, we've talked about that it will attach to the phagocyte and cause the phagocyte to open up and eat whatever the antibody is covering plus the antibodies. Okay, these are the things that are produced by those plasma B cells as part of the primary response, and by memory cells that are responding to a second exposure to that antigen, to the secondary immune response. It is the most prevalent antibody circulating in the tissue, fluids, and blood, and it neutralizes toxins. It opsonizes, right? So it covers those bacteria, and it also is part of the complement. So it helps to fix the complement on the cell membrane, which then it forms that hole in the cell's membrane, which allows all of the insides of the cell to leak out, and it kills it. Okay, the second most common antibody in the blood, at about 13%, is the IgA, which we just talked about. It comes in a monomer as well as a dimer form. Okay, and it's about 13% of total antibodies in the serum, has an average half-life of about 6 days. This and every other one that we're going to talk about does not cross the placenta. That's pretty much exclusively IgG. And the dimer is the secreted antibody on mucous membranes. And the monomer is found in smaller amounts in the blood. Okay, the next one is IgM, and that's a pentamer. So there are basically 5 immunoglobulins that come together to form a ring, and that's the IgM type. It's about 6% of the total antibodies in the serum. Average half-life is 5 days. Again, does not cross the placenta. Now, the IgM is the first antibody in a primary response, in a first response to exposure to an antigen. And also, it can serve as a B cell receptor. So the individual monomers of an IgM can function as a B cell receptor. It's not the primary one that does that, but it can if necessary. The primary B cell receptor is the next one, the IgD monomer. Okay, and that's about 1% of total antibodies in the serum as monomers floating around as antibodies. If it's a B cell receptor, obviously it's not considered an antibody, it's attached to the B cell as a receptor. Has an average half-life of 3 days. And again, this is the triggering molecule for B cell activation. So if the antigen hits that IgD, okay, and it's attached to the B cell as a B cell receptor, that's the one that starts the clonal expansion and all of the other activities that happen when B cells are come in contact with the antigen that it's designed for. And the last one is IgE, and it's a little bit different. As you can see physically, it's a little bit longer. There's a little bit more length on that FC area. And basically the big difference is that it does not bind to phagocytes. It bonds to those mast cells and basophils, which are involved in the immune response. So, I'm sorry, involved in the allergic response. So this is the allergy antibody. It also addresses helminth infections. So this is the one that goes after worms. But the biggest thing that it does is it mediates anaphylaxis, which is a very severe allergic reaction. We're going to talk about this in the next chapter. And it also mediates things like asthma and other things that can be triggered by allergies. Okay, so there is a term called a titer, which is the level of antibodies in the serum over time. Okay, and this is a well-accepted principle that memory B and T cells are only created from clones activated by a specific antigen. However, there's new research that suggests that exposure to a particular antigen can result in memory cells that are chemically related to that antigen, even if those antigens have not been seen by the host. Okay? So in that case, the activation of specific memory occurs via recognition of epitopes, of antigenic sections of a a pathogen or something like that. And what that does is it allows us, instead of having to have the full pathogen, you can use pieces of a pathogen, or you can use non-pathogenic microbes in vaccines that will then give you protection against the more dangerous ones. We often refer to that as an attenuated strain. Okay, if, for example, in the MMR vaccine, those are all viruses, but they are attenuated strains of those particular viruses. So they're not powerful enough to make you sick, but they look enough like those 3 viruses that we're able to mount an immune response to get immunity against those particular viruses. Okay, so this slide talks about the very first time that you are exposed to a particular pathogen, or at least the antigen, the epitope that the immune system recognizes when you're exposed to that. Okay, so over on the very left, you see a little tick mark on the x-axis, okay? And that is the first exposure ever to an antigen. You then have a latent period. Okay, and again, this is looking at the numbers of antibodies in the bloodstream. So that latent period is actually when the innate immune system is kind of taking care of business. So the phagocytes are discovering where the pathogens are, they're eating them up, they are digesting them and putting those antigens out on their cell surfaces with the intention of doing a handshake with a helper T cell once it finds the one that interacts with that particular antigen. So that takes a period of time. And at this point, the pathogen is winning. We haven't been able to mount much of an immune response yet. They are growing like crazy, okay? But sooner or later, the handshake happens, the immune system wakes up, and we are able to mount that proper adaptive immune response, okay? So again, we talked about IgM being the first antibody that is produced. So that's the red line. So if you take a look at where the big blue line, the total antibody count, really starts rising, the very first bump you see is in that IgM. Okay, and then as IgM is trailing off, IgG is taking over. Okay, and so basically once you get that big IgG bump, that's when you're starting to basically take care of the infection. Okay, so sooner or later the infection comes back down and we're able to slow down that immune response. So this is where those regulatory T cells kick in and say, okay, infection's under control. We don't need to be making a ton of T cells. We don't need to be making a ton of B cells. We don't need to be producing all these antibodies. So it basically shuts the immune system back down. Okay, so then you have some period of time between exposures. So let's say 6 months later, you get exposed to the same antigen that started this process off. However, at this point, you now have memory cells that have been created during that primary response. And because you have those memory cells ready to go, they're ready to divide. They are ready to produce antibodies. And so as soon as you're exposed to that antigen, the handshake is going to happen virtually immediately. Those number of antibodies are going to shoot way up. And you can see that the number of antibodies produced in that secondary response is much higher than in the end of the primary response, okay? So the secondary response is faster and stronger to that same antigen. And that's kind of the whole purpose of vaccination, is to get you into that secondary response. So when you actually come in contact with the real pathogen, you're in the secondary response as opposed to that primary response. That's the whole point. Okay, so now we have a concept check. We're going to match each antibody type with its description. Okay, so let's start with number 1, IgG. And the answer to that one is D. It's the most prevalent antibody. Number 2, IgA. And that's the one that is secreted on mucous membranes, so that's letter A. Number 3, IgM. So that one is the first antibody produced. Number 4, IgD. Okay, that's the one that becomes the receptor on B cells. And number 5, IgE. And that's the allergy one, so that's letter C, produced in response to allergies. Okay, let's move on to section 13.6. So now we're going to be talking about vaccination. So we're going to list and define the 4 different descriptors of specific immune states. We're going to discuss the qualities of an effective vaccine. We're going to name the 2 major categories of vaccines and then the subcategories under each of those 2 major categories. And then we're going to talk about the principle of herd immunity and the risks that happen when herd immunity is not maintained at a certain level. Okay, so there are 2 basic types of immunity. One of them is referred to as natural immunity, and that's one that's acquired through the regular biological experience. In other words, when you get sick and you develop immunity because of that, that is natural immunity. So in my case, I had chickenpox as a kid. I am naturally immune to chickenpox. Okay? Anybody who was vaccinated against chickenpox has artificial immunity to chickenpox, and that's protection from infection obtained through medical procedures like vaccines or an immune serum. Okay, so active immunity occurs when the individual receives whatever the stimulus is, whether it's exposure to the actual pathogen or exposure to the antigen through a vaccination. Whatever it is that activates those B and T cells to produce those immune substances like antibodies. Okay? Active immunity is going to create memory that renders that person ready for that secondary exposure chart, okay? Bigger, faster response. It requires several days to develop, so you have that whole latency time when the innate immune system is trying to wake up the adaptive immune response. So it takes several days. And when you get those memory cells from an active immunity situation, it should last for a relatively long time. That varies from pathogen to pathogen. Some of them, if you get it once, you are done. You know, one and done. There are others that wear off after a period of time, particularly if you receive that active immune situation through a vaccine. So there are some vaccines that require multiple doses at the beginning, and then there are other vaccines that require booster doses. So tetanus is a good example. You need a new tetanus shot every 10 years and every 5 years if you're in a situation where you're most likely to be exposed. So if you work on a farm, every 5 years. Um, the active immunity can be stimulated by either natural, like you get sick, or artificial means, like you get a vaccine. Okay, passive immunity occurs when an individual receives antibodies from someplace else, either a human or an animal. The recipient is protected for a very short period of time, even though they have never been exposed to the antigen. Okay. However, this is not the kind of situation where they're going to get any kind of memory for that antigen, okay? Because they have never produced their own antibodies. They've never gone through that whole cycle, which would lead to the production of memory cells, okay? So what you're getting is immediate protection and limited time, okay? So it's a short-term effectiveness. And again, this can either be natural or artificial in origin, okay? So again, this would be like you have been exposed to tetanus, you don't have a tetanus shot or it's not up to date, so they're going to give you some anti-tetanus toxoid to keep you from getting tetanus, which is a pretty devastating disease. So you definitely don't want to get that disease. So it's better to get those antibodies exogenously rather than risk having permanent damage caused by tetanus. Okay, so here are those 4 types of immunity. You can have natural immunity, which is either you get sick and you get better and then you have some level of immunity, or you can have that temporary immunity from something like colostrum or breast milk, where you are acquiring those antibodies from the mom who is actually producing them. Artificial immunity is either a vaccination, okay, so that's going to cause your own body to respond and create memory cells, or that toxoid that we were just talking about, where you're receiving antibodies from another source. And it gives you temporary immunity, but you're not ever developing those memory cells. So you are not immune at this point. Okay, so a little history of immunization. So the very first recorded attempt to immunize occurred in 6th century China. And basically it consisted of drying up smallpox scabs and blowing them with a straw into the nostrils of other family members of the person who died from smallpox. Sounds totally gross. Hopefully it worked, because that would be a really bad thing to get smallpox scabs blown up your nose and then get smallpox anyway. That would really stink. Then there was another attempt around the 10th century, and this was called variolation. And it was the deliberate inoculation of dried pus from smallpox pustules of one patient into the arm of a healthy person. And this method was used in parts of the Far East until it was brought to England in 1721. And the bad news is that didn't work very well, because many of the recipients still died of smallpox. Now, I guess if you're in a family and the people all around you are dropping dead from smallpox and it's very likely that you're going to die, maybe it's worth the risk. But wow, what a horrible, horrible idea. Okay, so comes Edward Jenner, 1796, and he basically hypothesized that all of those milkmaids who are well known for their beautiful smooth skin, because they seemed never to get smallpox, they got a different version of that pox virus called cowpox, and they came down with a few little scabs, but it never seemed to make that same scarring, especially on the face, that smallpox would do. And so he hypothesized that cowpox would make you immune to smallpox. And he then tested his theory by injecting a young boy with human cowpox, and then exposed him to smallpox 2 months later. And the boy survived, which is great, but it violates every single uh, uh, tenet of ethical research in modern day times. And I can't believe that even in those days he didn't see something fundamentally wrong with that procedure. But the bottom line is that because he did this, vaccination, while originally, you know, looked at with great suspicion, ultimately, because it proved successful, allowed vaccination to be accepted into modern society. So, vaccination is defined as any immunity obtained by inoculation with selected antigens. Okay, so the first attempts for passive immunization uh, is where you're actually taking um, horse serum with anti-tetanus antitoxins. Okay, so horses are constantly exposed to tetanus, and so they have developed an immune response to that. And so they took horse serum and they basically concentrated it down and pulled out the antibodies and were able to prevent tetanus and to treat diphtheria. Okay, we don't use that anti-sera from animals anymore. We now use human products. So we use immunoglobulins extracted from the pooled blood of many, many donors, and the process concentrate those antibodies to increase the potency, and also its process to eliminate any potential pathogens like viruses like hepatitis B and HIV. Um, and these sera are used by patients who have a diminished ability to mount their own immune response. Either they have some kind of a genetic defect or they have some kind of um, like HIV, where it suppresses the immune system. So any kind of immunosuppressive situation. In some cases, anybody who's had an organ transplant has been put on drugs which suppress the immune system. Okay, so basic principles behind vaccination is you use whatever components you need, antigen and other components of the vaccine, needed to stimulate a primary response leading to a memory response. So all of that primary immune response, all the time it takes to do that, and the lower response is bypassed. So it's done using an antigen that doesn't make you sick, but it primes your body, so when you come in contact with the real antigen, you mount a fast, strong antibody response. And then, of course, vaccines have profoundly reduced the prevalence and the impact of many infectious diseases that once upon a time were not only common, but were also quite deadly. Okay, think of polio, think of smallpox, think of even measles. Okay, so vaccine research is a huge, very hot area of research at this point. So what researchers are looking for are essentially these qualities of an effective vaccine. Okay, so obviously the first thing is it has to protect against the pathogen that it's targeting. It needs to have a low level of adverse side effects or toxicity. In other words, you don't want it to harm the person while you're trying to protect them. You want it ideally to stimulate both an antibody or B cell response as well as a cell-mediated or T cell response. You want long-term lasting effects. In other words, it produces memory cells. And ideally, it would not require a whole bunch of doses, and it wouldn't require booster shots throughout life. And of course, you want it to be inexpensive. You want it to be able to sit on the shelf for a really long period of time and make it easy to administer. Okay, so that's the ideal vaccine. Okay, so we have various different ways of preparing vaccines in order to build that memory, those memory cells for extended immunity. So you can have whole cell or whole virus vaccines, which usually include live attenuated viruses or live attenuated cells. So an example of that would be the MMR vaccine, measles, mumps, and rubella. And all of those are attenuated viruses. You can also use killed cells or inactivated viruses, which can still produce the immune response. Okay. Um, You also have the possibility in some cases of just simply extracting the antigenic regions of those pathogens and putting that in a vaccine. So really the point is that you are exposed to the antigen in a way that wakes up the immune system. So if you can do that with just pieces of the bacterium or pieces of the virus, that's much better because that has virtually no possibility of making you sick. So you can get subunits derived from cultures. You can synthesize subunits to make it look like natural molecules. You know, if you get an immune response from something totally made in the lab, you know, what you're looking for is the immune response. So you can get genetic engineering involved in that. Um, the other thing that often has to happen is that vaccines need to be conjugated. And what that means is that you take the antigen, which may be really, really small. And remember, the size of the antigen is involved with how antigenic it is. So here we take that antigenic region, which may be fairly small, and we stick it onto a protein. So the protein is much bigger. It's going to increase the antigenic response, but the antigen itself is what it's going to be responding to. So it just makes the whole thing more immunogenic to make a bigger immune response. Okay, so here is basically a picture of what we just talked about. So we have whole cell or whole virus vaccines. Typically those are attenuated, so they do something to make them less virulent, but still have the same antigens on the viruses to actually make the immune system learn how to fight off that particular virus in the wild type. Subunits are basically where you chop things up and use pieces in order to get that antigen, the response to that antigen. Okay, there's a fairly new type of vaccine, which is microbial DNA inserted into a plasmid vector and inoculated into a recipient. Some of the human cells will take up some of those plasmids and express the microbial DNA in the form of proteins. Okay, and those foreign proteins will be recognized during immune surveillance and cause B and T cells to wake up and form memory cells. Okay, I'm not sure how crazy I would be about getting a vaccine like that, but I will wait and see what the literature has to say. Okay, so most vaccines are administered still using a needle, using an injection, either subcutaneous, intramuscular, or intradermal. Okay, so still it's a shot somewhere. Um, there are some, not many, but there are some organisms that can be done either using a nasal or an oral vaccine. So for nasal vaccines, there's the flu mist, which is given to a lot of school-age kids by the school nurse, and it's just like a little nose spray. So you just squirt some of the vaccine up the kid's nose, and they are vaccinated against the flu. Oral vaccines for many, many years. And when I was a kid, this is how I got my polio vaccine. They put something on a cube of sugar and popped it in the mouth. And, you know, kids are pretty receptive to getting a sugar cube, even if it's got a little polio on it, you know. It's much easier than giving vaccines, and the kids are perfectly happy to take it, whereas they're not so hot on the idea of having this sharp thing coming towards their arm. So there are also a number of research groups that are working on developing a vaccine delivery system using a transdermal patch. So you would take your vaccine on a patch, you would put it on your arm, you'd leave it there for some number of days, and that would be the vaccine as opposed to something sharp going into the arm. There's another component to most vaccines called an adjuvant, and it has a number of different properties. This, first of all, enhances the immunogenicity of the antigen. So it basically makes it more antigenic. It gives you a bigger immune response. It also sort of ties the antigen to the injection site to give a high enough concentration for macrophages to go and find it. It precipitates the antigen and holds it in the tissue so that it can be released gradually. Okay, so again, it just makes it a little sticky. It facilitates contact with those antigen-presenting cells like macrophages and dendritic cells, which are going to actually start the process of making those memory cells. And the most common adjuvant is an aluminum salt called alum. Okay, so vaccine side effects. So vaccines, like any other drug, have to go through a whole series of steps before they're released to the public. So they go through trials in animals, and then they have human volunteers, and it's a while before they're licensed for general use. Typically, the complications that are recognized as part of vaccines are local reactions. So you might get a huge red welt because you have some kind of a localized allergic reaction. You can have fever. You can get, like I say, an allergic reaction that may be more systemic. But most of those serious reactions are pretty rare. So one of the really important things about vaccine um, usage is that we have this concept called herd immunity. And essentially, if you can get a certain percentage of the population to be immune to something, that pathogen can't get a foothold in that population to be spread. Okay? So herd immunity essentially breaks that pathogenic cycle and stops the transmission. And so if you don't get that level of herd immunity, which is usually somewhere between 85 and 90%, then you're going to start getting outbreaks. Measles, mumps, diphtheria, polio, typhoid fever, whooping cough have popped up various places, often in places where people live very closely together. So college dormitories are a really big way, a really big place for these kinds of outbreaks to happen. Also airplanes. Um, definitely, when people are just really close to each other, if one person is sick, it's pretty easy to pass it to the surrounding people. Some religious communities, for example, the Amish, do not vaccinate their children. And they in fact are one of the few places where measles is still pretty common, and they have measles parties. I told you about the chickenpox parties that used to happen when I was a kid. Well, they have measles parties to, get the kids sick all together and at about the right age, and they all get measles, and then, they're done. Um, so again, I said you have to hit that certain level of herd immunity in order to get population protection. Okay, so concept check. Determine which of the statements below describes active or passive immunity. Okay, so the first one is infusion of gamma globulin. Okay, and that would be a passive immunity because you're taking antibodies from somebody else or some bodies and injecting them into a person who either can't make their own antibodies, or by the time they were able to do that, they would be dead from whatever disease it is. Recovering from flu. That would be active. So you got the flu, your immune system dealt with it. That's an active type of immunity. Receipt of the influence of vaccination. Well, that would be passive, okay? Because you're receiving the antigen and you are making your own immune response, but it is provided passively, artificially. Antibodies passed from mother to infant through breastfeeding, and that would be a passive immunity, receiving the antibodies.